1E and 1F. I'm going to do a quick recording on the practical that we did last week. I hope you have your practical book with you so that when I'm running through, you can record down the answers and listen to some of my explanations that I'm going to give. Okay, I think the first experiment 6.1 we did on basic usage of a microscope. Basically, you know the purpose of a microscope uh, is to actually observe different kinds of cells, something that we can't see with our naked eyes. Okay, then we started off with this letter E. The letter E was facing you, okay, when you place it on the microscope slide. So this is how it will look like when you look through the lens. So you can see it is laterally inverted and it's also vertical. Okay, so if you have not drawn this, can you quickly draw this on your booklet? Okay, so I hope you know what is the meaning of laterally inverted. So if I raise my right hand, if you stand in front of a mirror, so the left hand will be raised. Okay, if you move towards your right, so it will look like you're moving towards your left. Okay, now if you move towards your left, the direction will be to your right. Can you just write it down? And if you move the slide towards you, the direction will be the image will move further away from you. So it will be away from you. Okay. Let's try this few discussion question. Okay. The first question is why should we never touch the lenses with your fingers? Okay. And these lenses should be cleaned with special lens paper. Okay. What is the reason for this precaution? Okay, this is the reason. Okay, touching the lenses with your finger or whether a cloth may scratch the lenses because they are precisely constructed. We usually use lens paper to clean the lenses to prevent leaving fingerprints, damaging or scratching the lens. Okay, that is why we use a special lens paper which is designed to clean the lenses. Okay, we should not be using your fingers of water or any kinds of towel, so there might be some scratches and you might not be able to see the image clearly. Okay, question two. This boy Jimmy, he's trying to conduct some experiment and he did some minor changes. Okay, the first one, he took two glass slides A and B and he cut out the letter E. And the first one, he put a cover slip. I hope you remember the cover slip. That's the one that many of you broke that day accidentally. It is very thin, okay, on slide A, but not on slide B. Okay, and what is the purpose of Jimmy? What was Jimmy trying to find out? Okay, basically, he wants to see how the cover slips affects the clarity of the image. Okay, under the microscope. So how does this E that is placed under a cover slip look different from the one that is placed, the slide that is placed on B? Okay, and he also observed the cutout of slide A is clearer than the cutout of slide B. Does appear to be clinging to the cutout of slide B, which was not covered with the cover slip. Based on this, what can you infer about the purpose? So basically, why a cover slip is necessary. Okay, the cover slip is used on slide A but not on slide B. Um, it gives you the clarity because you're placing the cover slip on top of the specimen, so you're pressing it down so light can shine through clearly. Okay, so it makes a difference and then it blocks off from dust particles or even the specimens from being blown by the wind and so on. Okay, the cover slip, it press the specimen flat. Okay, and what is the purpose? It prevents the specimen from being blown by the wind. Okay, and also protects the specimen from dust. Even the water particles, sometimes we add iodine or water to give the clarity, right? So we don't want any water bubbles from being there. So when we place it down, we are pushing the bubbles out, the air bubbles out. Prevents water air bubbles as well. So cover slips are important when we want to view a specimen via the microscope. Okay, that's for experiment 6.1. Basically, the usage of a general microscope. Okay, let's move on to 6.2. I know we did not do this experiment because of we don't want you to scrub your cheek cells. But whatever the case is, a cheek cell, it's an animal cell. So we have learned this is how an animal cell will look like. Very irregular. Okay, if you view your cheek cell via a microscope, the nucleus will be very clear, cytoplasm and cell membrane. Okay, something about drawing the structure. This is biological drawing. When you do biological drawing, the lines that you show to label must be a straight line. Okay, and you don't want to draw in this manner where you place the labels everywhere. So this is not a good drawing. So I'm going to draw another drawing by the side. So probably you just want to follow. So this should be the one. We have the animal cell. This is your cell membrane. Okay, and this is your nucleus. So maybe I shade. Okay, how do you label? So maybe you want to label all on one side and it has to be straight line. If there's no space, I'm going to do this. So if you're going to slant, this line has to be a straight line. Then you put cell membrane. Okay, this is a requirement for biological drawing. So you cannot draw slanted line like this. Not allowed. Okay, and we want to place all the labeling on one side. So this one has to be your nucleus. I got no space, so maybe you just want to add on. Okay, and then you have cytoplasm, no space. I'm going to draw like this, again straight line. Then you write cytoplasm. I got no space, so maybe you write it on your own. Okay, so your labels all on one side. It can be this side or this side, and there should not be any shading. It should be a solid line. Okay, so this is your biological drawing. So maybe you just want to amend for this. Okay, done. Okay, let's try. State the parts of the animal cell shown in the figure above. You can see all these my small dots. I think you know they are all vacuoles. And inside the nucleus, you have all this thread-like substance, which is your chromosome. Okay, so this two. So write it down. Okay, then the next cell, we did not give you a liver cell. So liver cell is also another specialized kind of animal cell. Okay, so this is an enlarged. So these are all the common ones. Again, not so right. So can we quickly draw nucleus? This one. Okay, here cell membrane. This one will be your nucleus, and here will be your cytoplasm. Okay, you can see my lines are all straight lines, and my labels, the names of the parts come in one side. If you want, you can also write it at the side. Okay, can you just draw it? This is not so right, so maybe I just remove. Okay, biological drawing. Okay, let's run through some of the discussion question. The first one is why we are using iodine. Okay, for the first one, if you remember, probably not for this experiment, we didn't do this experiment. For the plant cell, when we asked you to take out the epidermis of the onion cell, we actually asked you to do put a 1 drop of iodine. Okay, because the epidermis looks like a colorless layer, so it's going to be difficult for you to see the lines and all that. Okay, basically the iodine solution highlights the different parts of the cell. so you can observe more clearly under the microscope. Okay, basically it stains, okay, the different parts, and it highlights the details. Okay, it stains the different parts and it highlights the details. Okay, so you can write it down. Okay, when we scrap the liver cells, we do not take thick scrapings, usually very thin layers. Okay, explain, predict what will be observed if I use thick ones. Okay, you will see the image as very dark. Okay, the reason behind the light might not be able to pass through because it's too thick. Okay, you want to get a clear image, a lot of light must be able to pass through the specimen. Okay, if it's too thick, then there will be lesser light passing through. Okay, so the thicker it is, it hinders the passage of light, and a dark image will be seen. Probably we'll see a bit of image, but it will be dark. Okay, that is question 2. Okay, question 3. Lucy observed a liver cell without knowing what it was. But how does she know it is an animal cell? Okay, one of the ways is you will have nucleus as the largest part of the cell, and there will not be any cell wall. Okay, probably not so regular. Okay, maybe you can say it will be a little angular in shape. Okay, it's not very regular, like rigid. Okay, question 4. The nerve cell and red blood cell will look different. Okay, it definitely will look different. Maybe I can show it to you when you come back to class, or you can view it in Google or your textbook. There's red blood cell looks really different. Okay, a bit of concave in the center. Okay, because they have specialized functions. Okay, because it has to meet the function, it is designed in that way. Okay, biconcave in nature and so on. Okay, because the purpose of a red blood cell is to transport oxygen to the rest of the parts of the body, so the shape has to be such. Okay, so the surface area is we are increasing the surface area for oxygen to cling on. So probably that is why the shapes the appearance looks different from the normal cheek or liver cell. Okay, that is 6.2. Okay, I know I'm not giving you time to copy. It's a recorded video, so you can pause while you're writing down the answers. Okay. Okay, this was the last experiment 6.3. I know it was a bit rush. Okay. Um, okay, we used the epidermis of an onion cell, and then we actually dropped iodine to give you the clarity so that it actually stains the different parts. Okay, and large view, we are supposed to draw and label. Okay, again, labeling all wrong. It's not wrong, the way it labeled is wrong. Okay, the nucleus, can we draw a straight line like this and then put here nucleus? Okay, the cell membrane is okay. The cell wall is the outer one, is again slanted. So maybe you just want to put a straight line. Okay, can you remove this vacuole? You have a large central vacuole, so maybe like this, vacuole. And then the cytoplasm also we can remove. Okay, this cell membrane is cannot be going in. This is wrong. You remove, or it should be touching this line here. Okay, and your cytoplasm is the one that is inside here, so you write down cytoplasm. So I want to see all the names, the labels at one site. Okay, and you should not have slanted line. You can have slanted line, but this has to be a straight line, then you label accordingly. Just take note, the cell membrane is this inner one and cell wall is the outer one. So maybe I change the color. This is your cell wall, so this is the outer one. Okay, So the cytoplasm is slightly inside. Okay. Okay. Let's move on. The second one that you observed was a hydrilla leaf, right? Okay, we're going to draw and label the diagram. We didn't have any time. It was quite rushing. I think one thing was prominent for those who were able to observe the hydrilla leaf was the presence of the chloroplast. Okay, again, biological labeling. So I'm going to remove all this. We want to label all on one side. So probably straight line, then you put down nucleus. Okay, cell membrane is this one. Okay, this is straight line is okay. Cell wall, maybe another straight line. Okay, and your cytoplasm, if you got no space, you can draw a slanted line but straight. This is your cytoplasm. Vacuole, again, slanted but straight line here. And of course, all these tiny circles are your chloroplast. So straight line, put down chloroplast. So it is concentrated. The labels are all on one side and they all have straight lines. Okay, please ensure you label accordingly and not give me slanted lines like this and your labels are everywhere surrounding the diagram. Okay, so basically the extra feature will be a chloroplast which is inside the hydrilla plant. Okay, let's answer the question. The first one, state an organelle that is present. Organelle means parts of the plant that is present. So maybe just take note. In hydrilla leaf, but absent. I think this is quite obvious. Chloroplast, which is absent in the onion epidermal cell. Okay, just to take note, only cells from plants contain chloroplast, and onion is an underground storage root. Okay, storage organ. Maybe I write it down. Underground storage organ. Okay, so you will not see the chloroplast. Okay, its fleshy cells do not contain any chloroplast. Just take note. Okay, part B. Suggest the reason why this part of the cell in A is absent. Okay, it grows underground. Okay, do not require light. Okay, so we are not going to photosynthesize. We're not going to make food. So that is why these cells do not require. So no light required. It is more for storage and you do not photosynthesize. So do not need chloroplast. Okay. Looking at the drawings in 6.2, which is your animal cell and your plant cell, list three differences. I think you already know. Okay, a plant cell, you have a cell wall present. Okay, absent in animal cell. This is prominent. And then in plant cell, you saw chloroplast, and animal cell is absent. And in a plant cell, you have a large central vacuole, but in animal cell, you have tiny vacuoles. Okay, you also can talk about cell wall, okay, which is circular. You also say cell wall in animal cell is circular, for a plant cell, it's angular. Okay, or you can say rigid cell wall. In plant, in animal cell, there's absence of cell wall. Okay, that's all for the practical. Maybe you want to use the time to repeat and ensure the questions that I have highlighted are completed with proper biological drawing of the animal plant cell and bring the practical back to class next week. Okay, that's all for today. I will end off here. I see you all the next lesson. Probably the next lesson on Wednesday will be an SLS lesson. Okay, thank you and see you. Bye.